I wish you all a very warm welcome for this wonderful topic in this channel, and I wish all my viewers to have a beautiful and safe life in this pandemic time. Today we will be discussing about another chapter of biotechnology, that is about restriction endonuclease enzyme. In this video we will discuss up to naming of restriction endonuclease and the plane of cutting, that is the blunt end and sticky end, how these enzymes are generating it. First of all, we have to discuss what are their utility, what restriction enzymes are. Okay, let's discuss in detail. In biotechnology, it is now possible to isolate. Suppose this is one cell and it is another cell B. Suppose we want to isolate a gene from A and transfer it to B. How is it possible? Through isolation technique. It is a separate technique. I have described it in previous video. It was described already. After isolation of DNA, a specific genetic segment from it is isolated. The gene present in the DNA, suppose this is the gene, we can cut the gene from the DNA without delivering the total genetic material. We will cut the specific segment and have to introduce it. Okay. So, after isolating this segment, it is loaded to a vector. What is a vector? The vector is a carrier molecule, and in this time we are using plasmid as vector. So, the vector carry it and give it to cell B. The cell B became genetically modified. So, this cell became source cell, it became the host cell, the host cell became genetically modified cell. Suppose we have intended to produce insulin. So, insulin gene is isolated and introduced to it. In this, hence, it is the basic technique of delivering DNA. The question is that how to cut the desired segment? There must be a molecular scissor to cut it. This molecular scissor is nothing, rather it is the restriction endonuclease, and we will discuss detail about this restriction endonuclease. Clear? So, the restriction endonuclease. So, this restriction endonuclease is one of the most important tool in this biotechnological approach. Okay. We can obtain the gene from any type of cell from our body, because all cells have same type of gene. तो इसका कैरेक्टर क्यों अलग है सेल का? सभी सेल में एक ही तरह का जीन होगा, लेकिन the expression of genes are variable. So, all cells have variable functions. Okay. Suppose we have to collect insulin gene, we can take it from any cells of body. We do not have to isolate the beta cells from pancreas. Okay. So, let us discuss what are these restriction endonuclease first. Okay. Inside the cell, any type of enzyme which are used to cut DNA are called as suppose any enzyme cutting DNA are called as DNase. Any enzyme cutting RNA are called as RNase. Any enzyme cutting nucleic acid are nucleases. So the nucleases are divided into two different groups, DNAs and RNAs. The DNAs are again divided into two groups. What are they? These two groups are endonuclease and exonuclease. What is the difference between them? The endonuclease cut the DNA at any randomly at the mid region of DNA. Suppose this is the DNA. Suppose this is a DNA. When the DNAs cut it from when endonuclease can cut the exonuclease cut the DNA from the end sequence only, from the end. It cannot cut from the middle. Okay. So this is the difference between endonuclease and exonuclease groups. Then what are restriction endonuclease? The group of restriction endonuclease were discovered during 1963. एक अच्छा सा स्टोरी होगा एंडोन्यूक्लियस का, हम लोग पढ़ेंगे कैसे हुआ, कैसे आइसोलेशन हुआ रेस्ट्रिक्शन एंडोन्यूक्लियस का, उसका नाम कैसे दिया गया. लेट्स हियर. In 1963 the endonuclease were first discovered when the bacteriophages or virus attack bacterial cell. This is the bacterial cell. The virus attached to the host cell. After attachment, as per the nature, it transfer the genetic material. ये बैक्टीरिया का DNA. This is the DNA of host. This is the DNA of bacteriophage. When the DNA of virus entered into it, suddenly a group of enzymes became active inside the bacteria, specific enzymes, and they started degrading the viral DNA. Okay, inside the bacteria, these specific enzymes which degrade the viral DNA are called as endonucleases. Okay. The word restriction is given before endonuclease because they restrict the multiplication of virus. Is not it? The virus are prevented from multiplying inside bacterial cell. So they restrict virus from multiplication. So the word restriction is given. The second concept is that they have the ability of cutting at the mid region, cut randomly at the mid region. So the word endonuclease is given. So it is restriction endonuclease. This endonuclease cut the DNA at specific site, which is called as restriction site. Okay. The restriction sites are also randomly present in the DNA. Okay. It cannot cut the DNA by itself. Always the cut is specific at a specific site. Then what should be a restriction site and how these enzymes are named? Okay. Restriction site, come to the restriction site first. Recognition site. The recognition site is always a palindrome. Do you know palindrome? Palindrome means क्या होगा? बेस सीक्वेंस हम लोग 5 से 3 तक जाएंगे। जो जो बेस होगा अदर स्ट्रैंड में भी वही वही बेस होगा 5 से 3 तक। ओके, सपोज वी कैन सी हियर 5 dash end 3 prime 3 prime 5 prime G G G C C C G G G C C C. Carefully watch it. The sequences from 5 to 3 are similar to the sequence here. Okay, so these are the palindromic sequence, and always the restriction endonuclease identify palindromic sequences. Without palindrome, it cannot able to cut. So why these are named? These are named according to these are obtained from E. coli, Escherichia coli. When the restriction endonuclease are named, the one letter is taken from the genus, E is taken from genus, then 2 from species, CO obtained from species, then the R stands for the strain, and the number is according to the isolation, according to the copy number, isolation number. कॉपी नंबर प्लाज्मिड का होता है आइसोलेशन का, ई को R1, ओके, ई को R2, ई को R3, ई को these restriction enzymes always got 2 different type of ends, blunt ends and sticky ends. What are blunt ends? Suppose this is a palindromic sequence, C C C G G G G G G C C C. The enzyme always got a specific recognition site. Suppose this is the recognition site, it clip here, so the two strands became this. There is base pairing between all these bases. None of the bases are in single stranded. Okay, only phosphodiester bond split here, so it is blunt end, it is blunt. Suppose we will take the example of E को R1, how it got? E को R1 here हम E को R1, it is specifically GAA TTC CTT AAG. It is a palindromic sequence, it is one of the palindromic sequence. E को R1 identify a recognition sequence between G and A here. So after cutting by endonuclease E को R1, it produces sticky end T okay TT C. Okay, सॉरी ये नहीं हुआ, एमती है C T T A A, and the other end consists of this G C T T A A. So it generate the sticky ends. Okay, the sticky ends are otherwise called as cohesive ends. The blunt end are not always preferred. The blunt end are not always preferable. Why? The blunt end cannot adhere easily. The sticky end have single stranded regions. The bases have a tendency to pair with other bases, so these can easily stick with each other. So we can choose the sticky ends rather than blunt ends. Okay. So in blunt end only phosphodiester bond splits, but what happen here? The phosphodiester bond along with hydrogen bonds, both type of bonds are cleaved by the enzyme. Okay. So all about this restriction endonuclease. In the forthcoming video we will discuss about restriction modification and type of restriction endonuclease. Okay. Thank you all.